Picture this. You flip open your Cambridge Biology paper 2, feeling totally confident. But what if a single, tiny missing word could literally drop your entire grade boundary? Well, it happens every single year. Students spend hours, days, memorizing textbook definitions, only to get completely blindsided by how examiners actually grade these multi-step biological pathways. Let's dive right into this explainer and make sure that doesn't happen to you. Here's a cold hard fact for you. Topic 2.2, glycosidic bond formation by condensation. It accounts for roughly 15% of your total marks in paper 2. 15%. Guys, that is literally the difference between a B and an A. And the real tragedy here? It's not that the biology is too hard. No, the tragedy is that candidates throw away easy marks on completely avoidable mistakes that examiners see on repeat. Just listen to this quote. It's pulled straight from the Cambridge Principal Examiner reports. The senior examiners are explicitly warning us. Students consistently confuse key terminology in multi-step biological pathways. This just proves that ordinary, surface-level revision, like just rereading your notes or staring blankly at flashcards, completely fails for this topic. Superficial breadth? Yeah, it won't save you here. So why do these multi-step biological pathways cost so many marks? Well, it all boils down to the assessment objectives. The Cambridge syllabus makes it super clear that top-performing students have to show a connected, conceptual understanding, one that you can transfer to unfamiliar contexts. If you just memorize a standard example of condensation, the absolute second the examiners swap the context or throws a weird experimental setup at you, that memorization just shatters. Suddenly, you aren't answering the question in front of you, you're just answering the textbook question you happen to remember. But hey, here's your first quick win, the method mark strategy. The assessment framework actually prioritizes analytical depth over superficial breadth, meaning examiners are actively looking to give you marks for your correct procedural analysis and scientific reasoning. Imagine you're extracting data from a graph on glycosidic bonds, interpreting it, doing the math. Even if your final calculated number is totally wrong just because you hit the wrong button on your calculator, you can still salvage most of the marks. How? Simply by showing your systematic analysis and evidence-based reasoning. Now, in just a minute, I'm going to reveal a huge secret hidden right in the principal examiner reports. It's a specific examiner clue that completely flips a low-scoring answer into a top-band response. But before we get to that, you need the foundational framework that actually makes that clue work. Because without this framework, even knowing the secret won't help you put it on the page properly. Section 1, the top-band framework. To shift from passive recall into systematic processing, you need this 4-step procedural sequence. This is exactly how you guarantee maximum marks for Biology 9700. Step 1. Identify the core biological concept. Step 2. Extract the specific evidence or data right from the stimulus materials they give you. Step 3. Apply clear scientific reasoning to connect that concept to your data. And finally, Step 4. Execute your answer using precise examiner wording. Top-scoring candidates show evaluative thinking by questioning assumptions and justifying their decisions with evidence, and they do it in this exact order. When the clock is ticking in the exam hall, applying this framework under time pressure is absolutely crucial, especially for data interpretation. So when you're tackling a question on glycosidic bond formation, your sequence needs to be rapid and precise. First, deconstruct the question. Second, extract relevant data from the tables and graphs. Third, select your analytical framework. Fourth, execute with clear notation and working. And fifth, present your final answer with the correct terminology. You've got to verify your results and format them exactly how the mark scheme demands. Section 2. Spot the error. Let's try a quick interactive challenge. We're going to look at a real-world attempt at a glycosidic bond question, and I want you to actively evaluate it. Take a look at this flawed student answer. Condensation forms a bond and releases water. Okay, I want you to literally pause this explainer for a second. Think about everything we just talked about regarding specific terminology. Why does this answer lose marks? What is missing? Go ahead, pause, and predict the top band answer. All right, let's see how you did. The top band answer reads, condensation between two specific monomers forms a glycosidic bond, releasing one water molecule per bond. Do you see the analytical depth there? The flawed answer was way too vague. The top answer names the specific bond, the glycosidic bond. It specifies that it happens between monomers, and critically, it notes that exactly one water molecule is released per bond formed. That kind of precise examiner wording secures maximum marks every single time.
Section 3. the top 3 costly mistakes.
These warnings come straight from the Cambridge Principal Examiner reports. These are the top 3 most frequent, frustrating errors that just drain easy marks on exam day. Number 1, candidates consistently forget to include units in their calculations for glycosidic bond formation. Number 2, they forget to double-check those units and the associated terminology before moving on. And number 3, which honestly is the most frustrating way to lose points, providing answers that just aren't specific to the context given. Like we mentioned earlier, spitting out a textbook definition when the question asks you to interpret a novel experiment, that's going to cap your score immediately. Now, before we get to that massive examiner clue I promised you, here's a highly actionable memory hack so you don't fall victim to terminology confusion. Create your own mnemonic using the first letters of key terms associated with glycosidic bond formation by condensation. It can be something as silly as giant cat's bite. Well, you get the idea. The weirder it is, the more likely it'll lock those definitions into your brain. That way, when you face a totally unfamiliar context in paper 2, you aren't struggling to recall the basic vocab, and you can focus entirely on your scientific reasoning and data extraction. Okay, here is that massive examiner clue I promised. This one single habit flips a low-scoring answer right into a top-band response. The examiner alert explicitly states, always define terms before using them in extended responses. Top candidates explicitly define key elements before applying them to unfamiliar contexts. Doing this immediately signals conceptual mastery to the examiner, and it virtually guarantees you those method marks even if your actual application gets a little tangled up later. Remember, define first, apply second. Section 4. The diagnostic cliffhanger. So, just to recap quickly, you now have the top band framework. You know the concept, data, reasoning, and wording sequence. You know to define your terms first to grab those method marks, and you know to avoid the trap of ignoring the specific context. But there is a massive problem that this strategy alone cannot fix. What happens if you execute this perfect framework, but you apply it to the wrong command word? Like, what if the question asks you to evaluate, but your brain processes it as explain? It'll instantly cap your grade no matter how perfect your biological knowledge of glycosidic bonds is. Misinterpreting these exam command words is the hidden cause behind hundreds of lost marks every single examination cycle. Because honestly, if you don't know the difference between evaluate and explain, does your biological knowledge even matter in that moment? That's exactly why your mandatory next step is to master those command words. In our next explainer, we're going to decode the exact biology 9700 command words that decide your mark band, so you never apply the right biology to the wrong instruction again. Make sure to subscribe to our YouTube channel, AC Exams, at ACE_IGCSE to excel in your Cambridge examinations. I will see you in the next explainer.